So let's talk about specific immunity and vaccination. There are ways to categorize how immunity comes about: natural versus artificial, and active versus passive. And there are basically natural, active, and passive. There are artificial, active, and passive. So we're going to talk about those. Natural immunity is acquired through normal experiences. Active, natural, specific immunity. You get exposed to a pathogen, it infects your body, your immune system fights it off. So you encounter the pathogen and your immune system actually has to activate. This does give us a relatively long immune response. It does help us to maintain that immune response. The graph that I just went through with the primary and secondary exposure, that lasts for a while. And if I keep getting exposed, I make more and more memory cells. My immune system gets better at responding every time I'm exposed. Passive, natural, specific immunity. With passive immunity, I'm not activating my immune system. In some way, shape, or form, I'm acquiring antibodies to something. In passive form, that is natural, that there's no medical intervention, that would be if a mother breastfeeds her baby, she is passing antibodies to that baby. Antibodies are passed from mother to child through breastfeeding. Now, the thing with passive immunity, it is not long lasting. Your immune system is not engaged at all. The ability to have memory cells and the ability to have that secondary response, that's gone. These antibodies will give you immediate protection, but they're short-lived. This won't last forever. It won't last for a month. It only lasts until those antibodies are destroyed. Artificial specific immunity is going to be medically induced immunity. Active is still pretty much the same as what we saw over there. I am going to inject you with a vaccination that has something in it that your immune system is going to react to. Did you get sick? No. But your immune system still mounts a defense, creates memory cells, and does everything that it would if you actually had the infection. That gives you a long-lasting immunity. However, with passive immunity, again, this is like antivenom, you are receiving antibodies to protect against an antigen of some type or a toxin of some type. Why would you do this? Timing. Yes, it is short-lived protection. It is not going to give you long-lasting immunity. But remember that in a primary response, it took 7 to 14 days for your immune system to get off the ground. If I've been bitten by a rattlesnake, I don't have time for that. In 7 to 14 days, the amount of tissue damage that will be caused by that venom is going to be enormous. So what do? I give them antibodies that surround it, just like I showed you the last antibody function. That FC region at the bottom attracts macrophages. They get rid of the toxin, and voila, I don't have to worry about it poisoning my body. However, when they are gone, they are gone. It's not like I can get bitten by a rattlesnake and all of a sudden I have immunity to rattlesnake venom. It doesn't work that way. Because of the time that it would take for your immune system to react, in those cases with the different types of venom that are out there, you do have to artificially give them antibodies so that there's very little damage done. Let's talk immunization and the history behind it. Two of the first recorded examples of immunization happened a long, long time ago. The first one happened in 6th century China. It consisted of drying and grinding up smallpox scabs and then blowing them with a straw into the nostrils of vulnerable family members. Variolation happened in the 10th century. There was a deliberate inoculation of dried pus from smallpox pustules of one patient into the arm of a healthy person. This was used in parts of the Far East until it was brought to England in 1721. Here's where things went bad. for a lot of the people that got this, they got smallpox and they died. This was not a safe way of doing it. In 1796, Edward Jenner was very inspired by a dairymaid who'd been infected by cowpox. This is what I was talking about earlier. Cowpox looks somewhat similar to smallpox as far as the virus goes. And a lot of dairymaids worked with cows, of course, because they're dairymaids, got cowpox. Cowpox is not lethal. Cowpox doesn't kill you. But once they were immune to cowpox, they were also immune to smallpox. And Edward Jenner noticed this. And I know you're going, okay, so what? The fact that he noticed this was a big deal because of what he did next. He tested the theory that if you had been exposed and gotten over cowpox, you were immune to smallpox by injecting a young boy, he was I think 8 years old, with material from human cowpox lesions. Then 2 months later, he exposed this little boy to smallpox. He never got smallpox. That data proved that he was immune to smallpox, even though it was cowpox that he had gotten sick from. The use of this vaccine was first regarded with fear and skepticism, but later it was adopted, and it was adopted because it was proved to be successful. Vaccination is any immunity obtained by an inoculation with a selected antigen. I know this doesn't seem very spectacular, but you have to understand how horrible smallpox was. Smallpox was awful. People were dying left and right. Having the ability to make them immune was huge at the time. Passive immunization. The first attempts actually involved the transfusion of horse serum that contained antitoxins to prevent tetanus and to treat diphtheria. Two diseases that create toxins were being treated with antibodies. Antisera from the animals has now been replaced by human products. We now have the scientific ability to make our own human antibodies. The problem with using animal a lot of the time, because their antibodies are slightly different than ours, it can actually cause an immune response in our body. Our own immune system recognizes that antibody is weird and will attack it. Having the ability to make it into a human antibody has helped. Gamma globulin, our antibody that actually gets made, is an immunoglobulin extracted from the pooled blood of a lot of human donors. Processing creates antibodies to increase the potency and eliminate potential pathogens. We don't take blood and then just give it to somebody else anymore. That stopped in the 80s after the HIV craziness. Now, when you donate blood, it's separated, it's filtered. Any extra thing that could be in there that could make somebody sick, we take it out. If you have somebody who is immunocompromised, this is also very helpful. It's not just about having antivenom available. Artificial active immunity are vaccinations. The basic principle behind a vaccination is we want to stimulate a primary response to create a memory response. We're priming the immune system so that if you get exposed in the future to a virulent pathogen, you already have an army established in your body. If the pathogen enters the body, the response is already going to be the response of a secondary exposure, not the first that takes 7 to 14 days. Vaccines have profoundly reduced the prevalence and the impact of a lot of different infectious diseases that were once very common and very deadly. The qualities of an effective vaccine. If I want to have an effective vaccine, if I want to create an effective vaccine, it has to have certain criteria to it. It needs to be able to protect against the exposure to both the natural and wild forms of the pathogen. You have to have a low-level adverse side effect or toxicity and not cause harm. I understand that there are people who go, well, I got vaccinated and I got a fever and I got... Did you get whatever the disease was? No. I don't want it to make people sick. I don't want them to be damaged by it. The goal is to prep their body for a natural infection. That's what my goal is. I do also want both B and T cell responses to be stimulated. I want it to be long-term. I want it to be a lasting effect. I also, if I can avoid it, don't want to have a lot of doses to it or a lot of boosters. Rabies vaccination is still, I think, like 20 shots. But would you rather get rabies or would you rather have the shots? It's kind of a cost-benefit analysis that you got to do. I understand that getting 20 shots is not fun, but at the same time, rabies is 100% deadly. So, yeah. We also want these things to be inexpensive and have a relatively long shelf life and be easy to administer. We can have wholesale or virus vaccines. These are going to have either live attenuated cells or viruses, meaning that they've been weakened so much that they can't make you sick, still expressing the same antigens, so your body still responds, but it doesn't make you sick. Or we can kill those things and then just put them in your body. The antigens are still present. Your body should still react to the presence of foreign antigens. Antigenic molecules derived from bacterial cells or viruses, the actual subunits, the pieces, can also be made from cultures, can be isolated, I should say, from cultures. The subunits that I'm talking about, you can make them via cell culture, whether from a bacterial cell or a virus. These pieces, these subunits can be made. They can be synthesized to mimic the natural molecules found in the pathogens. The subunits can be manufactured via genetic engineering. In other words, I don't actually have to have the bacterial cell and the virus. I can just isolate the one piece, and I can actually conjugate them to other proteins, making them more immunogenic. Remember I talked about the haptens and how they were very small and they didn't really create an immune response, but I could attach them to something else, making them bigger, and then the haptens would become the epitope. That's basically what they're saying here. I can attach it to something else so that it's more visible to your immune system and will cause an immune response. So looking at wholesale versus subunit vaccines. I just mentioned this. With wholesale vaccines, they're either dead or they're alive but attenuated. If I've killed them, if they're dead, they can't be infectious, but they still have their surface proteins. This causes your immune response. It activates your immune response. It can't multiply or do anything like that, but because it's a whole microbe with all of its antigens on its surface, it still stimulates that immune response without causing disease. A live attenuated cell or virus has been damaged so that its virulence is very, very low or non-existent. It is alive. It still has antigenicity. In other words, it still has antigens that we can respond to, our immune system can respond to. Something that is different with the live version is it can still multiply. This right here boosts the reaction of your immune system. Again, you get immunity from this, but you don't get sick, you don't get the disease. Comparing the two. Because these don't multiply, you're kind of limited in what you're able to create as far as an immune response. Having viable microorganisms that do this, that multiply and actually produce infection, but not disease, it causes a greater immune response in your body. With the live attenuated cells or viruses, you also get longer lasting immunity to this. It requires fewer doses. You don't have to get injected as often. So not only dose, but boosters as well. And this method is really good at inducing cell-mediated immunity. The catch. 1, they usually require really specific storage conditions. And depending on where this is going to, some places will have that and some places won't. Another disadvantage, there is always a chance that it can revert back to wild type. The hope is that it doesn't happen, but it is a possibility. Subunit, vaccines. If, as an immunologist, you are aware of exactly what the immune system attacks, you can artificially create that specific subunit, whether that is through cell culture or genetic engineering. Either way, you're creating this particular thing that the body reacts to, and you make your vaccine out of that. Because it is a foreign antigen, the body will react. Toxoids are a bit different. Pretty much you can alter the toxin that is being created slightly, but you still have, so that it isn't as bad for the body. So you've got this purified bacterial exotoxin, and you chemically denature it. So it's a protein, right? If I unravel the sweater, it's no longer the sweater, but the pieces are still there. Remember, I talked about epitopes and how with epitopes, you're talking about recognizing a piece, not the whole thing. Even though it's denatured, you're still initiating an immune response using the epitopes that are present, which then creates basically antitoxins in your body. It isn't the toxin that was originally there, but if that toxin shows up, I still recognize and have immunity to specific snapshots of that whole picture. The toxoid vaccines are used for things like diphtheria and tetanus and pertussis. DNA vaccines. Microbial DNA is inserted into a plasmid vector and inoculated into the recipient. Human cells will take up the plasmid and express the DNA in the form of proteins. These foreign proteins will be recognized during the immune surveillance and cause B and T cells to be sensitized and form memory. So here's my invader. Here's this genetic code. This is a protein sequence in its DNA. I'm going to isolate it and put it into a plasmid. Remember, a plasmid is a little circle of DNA that can be read. I inject my plasmid containing that little piece of DNA into a host. Their cells will then start making that plasmid, which is a foreign protein. MHC1 grabs it, puts it on the surface of the cell. It's recognized as something foreign, and it activates your immune system. How are vaccines administered? Most of us think of vaccines, we think of injections. So these can be subcutaneous, intramuscular, or intradermal. Under the skin, inside of a muscle, or inside your skin instead of under. More recent vaccines have been nasal or oral, but because of the route, you're only really stimulating IgA protection on the mucous membranes, and the number of vaccines that are available in this form are very small. The benefit, it's easier than injections, and a lot of people prefer this to getting a shot because a lot of people are scared of needles. Several research groups are actually trying to develop currently a vaccine that could be a transdermal patch. It's not there yet, but they're trying. Adjuvants. These are specific binding substances that are required for some vaccines to be effective. Pretty much these additions enhance an immune response. They make it better. They keep the antigen in place at that injection site for longer, meaning that it isn't all released at once. It actually precipitates gradually. What that's doing is it's causing your body to respond for longer. This facilitates contact with those antigen-presenting cells as well as the lymphocytes. The adjuvants are trying to make that immune response better to give you better immunity. Side effects. Remember that this is from years ago, because I'm going to mention something in a little bit, and I want you to remember this is from years ago. Most vaccines have to go through years of trials and experiments in animals and in human volunteers before they get licensed for general use. There is 10 to 20 years of research that's done before it even gets approved. There will always still be complications. There could be local site reactions where you got your shot. There can be fever, there can be allergies. There can be other adverse effects that weren't seen during the trial period. More serious reactions are usually extremely rare. Something you have to understand is I can test this on 10,000 people, but when it goes worldwide, you're not talking about 10,000 people anymore. You're talking about billions of people. Somewhere in that number of billions of people, there could be somebody who reacts very differently than the 10,000 that it was tried on. That's just life. That's just the way it works. But overall, because of how much is done, it's very rare that stuff like that happens. People have attempted to link childhood vaccinations to later development of things like diabetes and asthma and autism. I'm going to say this because it's the truth. The guy that wrote the paper regarding the MMR causing autism lied. That's a fact. He lied. A lot of scientists, when his paper originally was released, tried to replicate his results and could not get the same results. This is something that scientists do all the time. If you bring up something, they want to test it to see if you were telling the truth. He didn't. And the scientific community crucified him for it. The guy lost his medical license because he lied. And he admitted that he lied. There was never any supportive data saying that any type of vaccination caused any type of secondary disease. Vaccinations don't do that. That isn't what they do. Anyway, this is revoked. This is a lie. The price of not being vaccinated. For those people that have not vaccinated their children and they are around other children, there have been outbreaks of things that hadn't had outbreaks in years, like measles and mumps, diphtheria, polio, typhoid fever, even whooping cough. These have popped up all over the country, even at college dormitories. It's also fairly common to see it in anti-vaccination religious communities, also in airplanes. These diseases that were very low frequency are all of a sudden making a comeback because people are not getting vaccinated like they used to. There is a decrease in the level of herd immunity. Now, what is that? Herd immunity is a phenomenon in which a certain percentage of the population is vaccinated. And what that does, if somebody gets sick and everybody around that person is vaccinated, it can't go anywhere because everybody else has immunity to it. That's herd. However, let's say that everybody was still getting vaccinated and I'm a teacher and I have a class of 24 students and I get, measles. If everybody else is vaccinated, no one in that room is going to get measles. However, if 12 of those students, now remember, if I'm sick with measles, I can pass it to 15 people. If 12 of those people in that room are not vaccinated and I get measles, 12 people are going to get measles. When they go home, they're going to spread it to their families, because if they're anti-vax, I'm pretty sure most of their family is going to be anti-vax. If they go to church with a religious community that is anti-vaccination, yeah. You can see how herd immunity won't work if people aren't getting vaccinated. I am going to talk about the COVID vaccine while I'm talking about vaccinations, and I realize that this is not in your book. But it's not in your book because when that book was published, it didn't exist yet. So keep that in mind. What I'm going to talk about is what you need to know for your exam, but it will not be in your book. So let's talk about the revolutionary COVID-19 RNA vaccine. One of the biggest arguments that I have heard about the RNA vaccine that was developed for COVID was that it happened too fast. It only took a year. In reality, guys, the RNA vaccine, the idea of the RNA vaccine has been under development for 20 years or more. The reason that it never kind of gained traction or went anywhere was because there was a lack of funding. With most scientific research, if you don't have funding, you don't have a way to really develop your new techniques and things like that, because you can't buy equipment, you can't buy supplies. And COVID-19 happened, all of a sudden this drive started regarding getting a vaccination that would work against COVID-19. So all of a sudden they had this influx of money, and all of this research that had been kind of stagnant because of lack of funding took off. What they ended up doing was they took the mRNA code for COVID-19 spike protein. Now, we all know what a spike protein is. The spike protein is the key that is used by the virus to get into our cells. If we can attack that in some way, shape, or form, we can prevent it from getting into our cells. So they took the mRNA code right there from that spike protein, and they put it into this thing right here. You can also see it here. It's a nanoparticle that is made from phospholipids, the same thing that makes your cell's membrane. They added a few extra things. They added some salts and some sugars in the makeup. They added these salt and sugar molecules for protection, as well as to keep it intact long enough so that it could get into your cells. Because this is actually made up of the same stuff that makes up your cell membrane, it allows it to fuse with your cell's membrane and basically drop off this mRNA inside of your cell. The mRNA being dropped off in your cell is a big deal. Remember that your ribosomes will read mRNA and produce a product through the process of translation, which is exactly what happens. Once this little strip of mRNA is in my cell, the ribosomes in the cytoplasm will actually read this mRNA. They create the protein that is encoded within this RNA. And in the case of the COVID-19 vaccine, that's the spike. Remember how our viruses have spikes on their surface. The danger that most people complain about with this vaccine and changing our DNA or giving us infection or whatever can't happen. Think about it. If all I'm encoding is just the spike protein, does it have genetic material that can invade the nucleus? No. RNA only leaves the nucleus. 2, does it have the capacity to make things like the capsule? No, because the code for that is not included in this vaccination. As an infectious agent, just having a spike on my surface is not enough to do anything but what it can do, what my cells can do with this protein that has been made. This, I'll draw it like this spike that has been made. MHC1 that we've talked about in our T cell response can pick this spike protein up and display it on the surface of the cell that it invaded. As the antigen presenting cells consume the cells, they're going to, as their job is, display those foreign antigens. That leads to helper T cell activation, because remember, helper T cells
recognize MHC2 on the surface of antigen presenting cells. B cells are also being found, creating memory cells that react and release the right antibodies. Both my cytotoxic T cell, my helper T cell, and my B cell have made memory cells that are going to allow my body to react to this infection. Because this is most likely the same spike found that allows them into the cell, remember our example of how antibodies work where they bound to all of the spike proteins? If it can't get to its key, it can't get into our cells. The other thing that is kind of we're hopeful about, is that if this is what they're using, it may be very, very similar for every single COVID variant, meaning that it confers immunity to multiple variants. Now, here's the big kicker, this right here. The way that this works, I have a very basic nanoparticle that I can use almost like an envelope. I can put whatever I want into this nanoparticle. And if I inject it into somebody, it will fuse with the membrane of the cells in this person and deliver that mRNA, allowing my body to have the ribosomes create a foreign protein, which gets put on the surface of my cell. And voila, I have an immune response. Most vaccines take 10 to 20 years to develop because in the past, every vaccine has had to have a unique design to match whatever the illness happened to be. Here, in the case of how our RNA vaccine works, we literally can use the same basic nanoparticle here, and all we have to do is input mRNA into this space. As long as we know a target for our immune system to recognize and attack, we can create that and input it into this little bubble that we've created in this nanoparticle, and it'll follow the same route that the COVID vaccine did. It'll get input into our cells. The mRNA will get read by the ribosomes, create a protein. MHC1 will pick it up and put it on its surface and start the chain of events of our immune system. Instead of our vaccinations taking 10 to 20 years to develop, this could be developed within a month. And unlike the other vaccines, the thing that I'd need to be created novelly for every one of them, new for every one of them, would just be this little piece of mRNA, which in a lab, you could create enough volume of that within a couple weeks. This could revolutionize how vaccines are made. That's what is so mind-blowing here. Instead of taking years, it would take a month, maybe a month and a half. The only thing that would impede our progress would be finding the target for our immune system to attack. But once that was found, it's a very fast process to actually create these little particles. We already have the base. The only thing we would need to create from the ground up would be the little piece of mRNA that we need to put inside this bubble. I also wanted to mention something else that people commonly say regarding vaccines that isn't necessarily accurate. A lot of people go in and they get their vaccination, and when they get their vaccination, they automatically, well, last time I got my vaccination, I got sick. Not true. Did you get the disease? Normally it's a no. The symptoms that they're talking about, either having brief fatigue or their joints are sore, or even if they have kind of a short-lived fever, those aren't symptoms of the disease. Those are symptoms of the immune system. And they are signs that your immune system is actually doing its job. Remember, fever comes from the cytokine pyrogens. So pyrogens is getting released by some cells in your body, causing your body temperature to go up. The soreness is a byproduct of inflammation. I mean, I will say this, you're injecting something into somebody's arm. Of course, you're disturbing the tissues, and you're causing damage to some of the tissues with that needle. So yes, you would have soreness. That makes sense. As far as brief fatigue, it takes energy to make all of those cells for an immune response. So again, you would feel kind of tired, but all of these symptoms that people describe have nothing to do with being ill and have everything to do with your immune system kicking on and doing the job that it's supposed to be doing. The design of this vaccine makes it impossible for you to get COVID-19. And if this is ever used as a design model in the future, it will also be completely impossible for those vaccines to give you the illness. Remember what is included in the vaccine. Salts and sugars aren't going to make you sick. Phospholipids aren't going to make you sick. Whatever is encoded in that mRNA is not designed to make you sick. It's designed to jumpstart your immune system so that you can have long-lasting immunity. The only drawback that we have with the RNA vaccines, the only thing that is an impediment to RNA vaccines is their storage. This was an issue when the COVID vaccine first came to be, in the fact that we needed them stored at -80. The fact that they are nanoparticles and the fact that they are such simple nanoparticles, we have to have the proper storage for them so that they don't fall apart. Keeping that in mind, if we keep using this platform, this structure, as a way to quickly and easily create vaccines against new infections that may pop up, new pathogens that may pop up because of the outbreak of COVID-19 and the necessity of getting a -80 freezer, even in small town pharmacies, we now have the ability to house those new vaccinations, even if we didn't before.